The fungal infections can be broadly classified into environmentally acquired such as the Aspergillus and Cryptococcus, or endogenously acquired as Candida. That is because Aspergillus fumigatus grows as a saprophyte in environmental niches such as compost and soil, and patients become infected via inhalation of spores. In contrast, Candida albicans is a member of the human microbiota, and invasive infections typically originate from colonizing cells of the patient's own gastrointestinal tract. Now, we are going to discuss the pathogenicity of the major two causes of invasive fungal infections, Candida and Aspergillus. Their pathogenicity is mainly mediated via a number of virulence factors that facilitate adherence to the mucosa, increase their ability to evade host defenses, and lead to the production of tissue-damaging hydrolytic enzymes. Here is a figure that shows some of the mechanisms of Candida's pathogenicity that is mediated firstly by the adhesion to host cell, then invasion or the formation of biofilm on non-living cells, which remains viable for years. The attachment of yeast cells to abiotic cells, for example catheters, or biotic cells as host cell surfaces, can give rise to the formation of biofilms with yeast cells in the lower part and hyphal cells in the upper part of the biofilm. Additionally, the Candida's capacity for mucosa adherence and biofilm formation is an important virulence factor where it forms a dry biofilms on environmental surfaces and equipment in hospitals, and can remain viable for several months. It confers significant resistance to antifungal therapy by limiting the penetration of substances through the matrix, and protecting Candida from the host immune response. Now to Aspergillus, where the relatively small size of Aspergillus fumigatus canidia allows access deep into the alveoli. Moreover, it can grow between 37 and 50 degrees Celsius, therefore more resistant and has better thermotolerance than other Aspergillus species. Aspergillus also secretes various proteases, hydrolases, and elastases with different functions. These enzymes may activate different pathways which have a direct effect on the expression of the virulence-related attributes of Aspergillus fumigatus, SUP-A9A/SUP. To infect the host and cause disease, many medically important fungi invade normally non-phagocytic host cells, such as endothelial cells and epithelial cells. Host cell invasion is a two-step process consisting of adherence followed by invasion. There are two general mechanisms of host cell invasion: induced endocytosis and active penetration. Candida utilizes two different mechanisms to invade host cells, either by induced endocytosis or by active penetration. During the first mechanism, the yeast cells as Candida adheres to host cell surfaces by the expression of adhesins. This contact with host cells triggers the yeast to hypha transition and directed growth. Also, the expression of invasins mediates the uptake of the fungus by the host cell through induced endocytosis. While during the second mechanism, fungal-driven active penetration has been proposed to be facilitated by adhesion, physical forces, and secretion of fungal hydrolases into host cells by breaking down barriers. This process would require viable hyphae to be completed. Additionally, the secreted aspartic proteases, which are produced mainly by the Candida species and contribute to its virulence, have also been proposed to contribute to active penetration. Shown here is a figure illustrating how Candida is attached first to host cells and then either enters the cells with the help of invasins, passively, or by the proteases through active penetration. Now, we are going to discuss the human immune response against invasive fungal infections. Firstly, once the fungi invade the mucosa, the host response is mediated by innate immune cells such as macrophages, dendritic cells, and monocytes, and by soluble mediators such as complement or different peptides. It has now been demonstrated that the innate defense is quite specific to fungal infection. The response is activated by a limited arsenal of soluble and innate cell-associated pattern recognition receptors, which can recognize conserved structures of microorganisms called pathogen-associated molecular patterns. Well-known fungal pathogen-associated molecular patterns include proteins and polysaccharides such as monon, SS-glucan, and chitin. The most important soluble pattern recognition receptors in the immune response against Candida are C-type lectin receptors, and the one against Aspergillus infection opsonization is known as pentraxin-3. Development of adaptive immunity is shaped by APCs expressing high levels of MHC, co-stimulatory molecules, and soluble mediators such as chemokines and cytokines that drive T cells differentiation. In the end, we are going to summarize the main complications of the major fungi as follows. The untreated Candida infection carries the risk of disseminated systemic infections in which other organs can become involved, and may lead to sepsis. Intestinal candidiasis can occur as a sequel to oral antibiotic therapy. Bronchopulmonary candidiasis is a rare complication of pre-existing pulmonary disease. Septicemia, endocarditis, and meningitis occur as terminal complications in immunosuppressive and leukemic patients. In leukemia patients, systemic candidiasis presents as prolonged neutropenia and fever refractory to the antibiotic. Central nervous system involvement is a devastating consequence of disseminated aspergillosis and may manifest with seizures, or focal neurological signs from mass effect or stroke. Additionally, the late diagnosis of cryptococcosis may lead to long-term neurological deficits defined by the presence of ataxia, poor mobility, hearing impairment, vision impairment, seizure, or cognitive impairment. While pneumocystis pneumonia in HIV-infected patients also leads to respiratory failure and mortality if left untreated. In summary, the most common medically important fungi are Aspergillus species, which is transmitted by inhalation, and Candida species, which is transmitted by direct contact. Additionally, the pathogenicity of fungi is mainly mediated through different mechanisms such as induced endocytosis and active penetration. Importantly, the innate immune system is the main defense mechanism against invasive fungal infections. Finally, untreated or late diagnosed invasive fungal infections are associated with several fetal complications.